So we're going to run today a little different, where we don't have very many slides,
But we have a lot of paper.
but we have a lot of paper. Sometimes you guys' faces on your thumbnails are about the same size as they are on your cameras, and so I always have this momentary feeling of, like, you know the scenes in movies where people like pretend like they're there by putting up like a statue of themselves. It's like somebody's like snuck out, but they've left the simulacrum.
All right, so let's do this. Today, we're doing long COVID. We got 3 papers. The first one that we're going to do is about the it's going to investigate some hypothetical. Actually, now that I think about it, maybe we should do the pathology one first. All right, let's do the pathology first. That makes more sense. All right, so what does it look like? What does long COVID look like? Let's talk about that first. That paper is in Nature Immunology. It's called Pathological Sequelae of Long Haul COVID. The next one we'll dig into some hypothetical mechanisms about what's causing this long-term COVID, and then we're going to talk briefly about the retrotransposon paper, if only because the authors are indicating that it might be responsible for some of the positive testing in the absence of infection, which can play into some of the other stuff that we'll get into. Cool. So
you want to start with the BMJ?
I think so.
All right.
It's probably the one that we don't need to spend that much time on. I think that the mechanistic paper is more interesting, but this will at least get us roughly started into what long COVID is and its prevalence, what systems are affected, stuff like that. They start off and they tell us that there is the persistence of a constellation of symptoms for time periods varying from more than 4 weeks from symptom onset to symptoms that last more than 3 months. And there's some different guidelines that have been proposed for that. But essentially the idea is that you're symptomatic long after infection and it's not attributable to other diagnoses.
I mean, the paper is going to be talking about the specific sequelae are manifestations. They're sort of clinical, physical, medical symptoms. And it's talking about organ-specific presentations. And one of the things that they say here where, you have the organ presentations of SARS-CoV-2 are related to SARS and MERS. We haven't talked much about SARS or MERS because these are viruses that are related. They're not identical in their presentation. They have kind of largely faded from memory. And one of the problems with being able to compare between SARS-CoV-2 and MERS is that the body of literature on SARS versus SARS-2 or MERS versus SARS-2 is way different. And that is pretty significantly, it complicates matters of being able to compare between them. However, there's a report that they demonstrate protracted neuropsychiatric symptoms, sleep abnormalities, persistent impairment of pulmonary function, including reductions in diffusion lung capacity, which we'll come back to later, fibrosis, myalgia, functional disabilities such as reduced exercise tolerance. So there's this whole collection of things that people get after SARS and MERS that they also seem to get after SARS-CoV-2. The next question is like, okay, so you have these specific systems that are affected. The next question is, well, are all systems affected equally, or are all people affected equally? Is mild COVID the same thing as severe COVID? And this is where it gets to be a little, immediately, right off the bat, we're kind of getting into territory that seems poorly or difficult to fully elaborate on. Because they say that the severity of your initial COVID-19, your acute COVID-19, has been linked to post-COVID-19 syndrome in one large nationwide study. Okay, so there's at least one study that says that the worse your COVID is, the worse your long COVID is. And then in another review, although COVID-19 associated symptomology was more pronounced in people, that had severe disease. Individuals with mild and moderate disease still reported a wide range of symptoms after the resolution of clinical disease, which meant that it doesn't necessarily have anything to do with how bad your sickness is.
It's also worth noting, maybe this is too, it's going to keep coming up over and over, but these are observational studies. Many of them, they're asking people how they're feeling 6 months later. And the way that you ask somebody how they're feeling often strongly informs how they answer that question, which is why observational studies tend to get in a lot of trouble later on in terms of why two studies like the ones Dr. Benary was just talking about could contradict each other so poorly.
Well, it's also the fact that you're looking at different populations. So if you're looking, the first study looked at the U.S. Departments of Veterans Affairs. This is a very specific population in the world that you're looking at and then trying to make statements about. And we're going to talk about this more next week because you cannot draw perfect parallels between two different groups, especially if you have a group that's veterans versus the general population. These people's bodies have been changed by war.
Absolutely. And likewise, if I ask you, hey, have you been feeling better these last few weeks? I know you had a really rough bout with COVID 6 months ago. You're going to get a very different answer than you're like, I bet it's been really hard on you these last 6 months. How are you feeling? Right? It's like, it's wild how much the phrasing of these things matters.
Definitely. And then, so basically, in addition to the fact that they found that individuals with mild and moderate disease also had a wide range of symptoms, there was another group that showed that 60% of recovered patients reported persistent symptoms at 6 months, no matter how initially ill that they were. Though, however, it's important to say that 60% report persistent symptoms, but it doesn't say that they report severe persistent symptoms or that all of them report the same symptoms.
Or it's like, what is that question? Do you have any symptoms? Or is it, have you had a cough this week? Have you coughed this week? Have you had sniffles? Have you had trouble sleeping? Like, it's hard to tell.
It's like the same way that if you have a trial, for your trial, you would try to take anyone away from the jury that had a preconceived idea about how somebody should feel, act, behave, respond, whatever. And so if you have people that are primed to be like, well, I've had long COVID, or I've had COVID, and I know that long COVID happens in the aftermath, and I have had some coughing, and I have had some trouble concentrating, they report that because they're like, oh, well, they're tracking long COVID, and so I report this. But it's like
also like, what's the background? Like, if I just ask anybody on the street, have you had any flu-like symptoms or cold-like symptoms or stuffy nose or anything in the last 6 months? Like, what's that look like?
And so it's very unclear. And whenever you see a situation where there's these observational studies that have been gathered in one place and they all point in different directions,
and they conflict with one another, it does seem to suggest that there is some kind of bias that's happening in the evaluation of these things that prevents you from being able to really say much. By the way, these biases aren't necessarily, we're not like insinuating these are maliciously introduced biases. It's the hardest thing in the world to fight against in experimental research because we're all human and we all have particular curiosities, and it's very difficult to not have them come out in the way that we collect our data. All right. They also take a moment here to note the fact that acute COVID, which is what we call the initial illness, versus long COVID appear to be different illnesses. Because long COVID is not just an extension of COVID illness. It is a collection of new symptoms that are systemic. Whereas, they say here, so the most common manifestations of COVID-19 are on the level of the respiratory system, the gastrointestinal system, cardiovascular and neurological systems. Then you have the manifestations of post-COVID-19 syndrome, which are these sorts of... Actually, now that I'm reading this sentence again, think it's trying to say that it's somewhat nebulous. Fatigue, sleep abnormalities, brain fog, mood impairment, cardiac palpitations. They're not the same as like your O2 sats are falling or something like that.
Sure, yeah. So it's like I think that I think that this is poorly written because they say the most common manifestations are systemic and they list them, and then they say by contrast the most common manifestations of post-COVID-19 syndrome are systemic.
Just different systemics.
Different systemics here, and I do think that what they're trying to basically show is that you have this different cluster, which is not you have cardiac symptoms and respiratory symptoms, but the fact that they bin them separately suggests to me that those who have cardiac symptoms are not having related respiratory systems versus cardiovascular system being listed here in the acute COVID-19 category would suggest that it's this concerted recruitment of the entire system. Let's see. So some lung stuff does maintain in these patients with COVID-19. It may or may not correlate with prior COVID-19 severity. It's really interesting reading these papers because this is what a year ago the literature on COVID looked like.
Yeah, this is going to be one of the more difficult lectures in terms of saying something conclusive. And we had this happen to us when we taught this class right after COVID started.
Because we started teaching this class, I think, in September of 2020.
Right, and so it was towards the end of the semester, we were getting into this territory where there were not definitive statements that could be made about a lot of the immunology.
If I remember correctly, it was from the very beginning of the semester, actually.
Thank God, the students became less tolerant of it towards the end. I think people were just like, what is going on? But now we've actually answered a lot of those questions. If we could jump in our time machine and go back, we'd probably probably be a lot more interesting of a discussion. So this is the edge of what's known today.
Yeah, so there's a bunch of stuff here too that they basically talk about the whether or not it's related or not, and then they're sort of like, well, maybe the pulmonary stuff is related to the severity. It might be related to the baseline of how sick you were when you first got COVID, because if you're sick and then COVID washes over you and you get sicker, then you might never return to the sort of the already depleted baseline that you were at. You might end up at an even more depleted baseline. And that's kind of to be expected, because if you're weakened and then you encounter something that causes you more damage, that you would have this extended defect.
And they make a note here too of possible mechanism involving these long-term scarring effects, these fibrous bands that occur, radiological persistence, stuff like that, which the other paper is going to get into more.
Yeah.
All right, so then we have a table that outlines a bunch of these. So in this sense, according to this table, systemic means diffuse symptoms like weakness and fever. Organist, what would you call them? Organ-based systems? Yeah, they should have said organ-based or something like that. So you have respiratory, neuropsychiatric, musculoskeletal, cardiac, vascular, GI, and endocrine. And that covers most of the bases in the body, which is quite fascinating. But you're talking about tissue level damage, I think, is the hypothesis.
And you see some overlaps, right? So dyspnea is shortness of breath. So you get shortness of breath both in acute and post-COVID. Cough in both acute and post-COVID. You have less likelihood of pneumonia and ARDS and hemoptysis, which are basically super, super severe manifestations of cardiovascular problems, but you do have decreased oxygen saturation that's crossing the alveolar membranes, this like wet cough, your scarring doesn't go away. So there are detectable physical characteristics, right? These sorts of these very hard things that you can point to where you're like, well, this person's oxygen saturation is still perpetually low. That would seem to suggest that there's some kind of extended problem that's happening. In terms of neuropsychiatric, you're definitely getting a totally different collection of neuropsychiatric symptoms in the aftermath of COVID than you do during. Though there is still some overlap. What's interesting is that brain fog isn't listed as being a neuropsychiatric manifestation of acute disease. which to me feels, again, like a sampling error, because when you have the flu, you definitely have brain fog. Like I remember still being in high school and having the flu and having a temperature, I think it was like 103, 104, and having these very...
Just having a fever is a pretty delusional experience.
Yeah, like your brain stops working. And so there's definitely some difficulty of being able to tell.
Makes me think that has to do with how they're asking the questions or something more than anything.
Right. Something like post-traumatic stress disorder. It's like if you are having if you are encountering COVID and despite all of your protections, despite being super careful, despite being very worried about it, and then you still get it, it's definitely something that can cause severe and lasting harm. And we know that PTSD can have physical sequelae, not just neuropsychiatric ones. And so you start to see this kind of network of things branching out against one another. Yeah, so joint pain, like arthritis is basically the extension of joint pain. When you begin to have joint pain, you will end up with arthritis over time if it's inflammatory in nature. We know that this is an inflammatory disease, so it makes sense that you would get arthritis. Like myocarditis and cardiomyopathy and these like very acute diseases appear to be relegated just to the acute phase. In the long phase, you start to have these kinds of harder to diagnose conditions, right? So chest pain, but not to the level of myocarditis or cardiomyopathy. Chest tightness, same thing. You've fallen below the sensitivity of the scans that people do, and so you still have the symptom, but they can't bin it as myocarditis because there's some test that they do for myocarditis, like what is your level of C-reactive protein in the blood and how much inflammation do you have in the heart. If you fall below that threshold, then you don't have myocarditis, but you still have chest tightness and pain.
Yeah, I was going to say also there's even way before COVID, there's just constellations of a lot of these symptoms that didn't have a good disease to hang on them, which is quite interesting.
Mm-hmm.
I'd be really curious to see the analysis of those nebulous diseases, like chronic fatigue syndrome, the one I can think of off the top of my head.
Mm-hmm.
But there's a number of these just, you go to the doctor and the doctor can't find anything wrong with you, but you feel horrible. I mean, I'm sure all of us have had this happen at least once. So with the seroprevalence closing in on 100% around the world day by day for COVID, it's quite interesting that most people are going to fall into the bin of having had it at some point. And therefore everything, like how do you separate that out from any other chronic fatigue situation?
And I also think that people are really, really tired. I think that people are tired and they're sick and they have been really damaged by the last 2 years, whether that's because they've been super careful about COVID
or
and
7 years. Or just the last 10 years have been crazy in our country. The last 5-10 years have been very stressful and hard on a lot of people.
Yeah, I mean, we're going through a massive social industrial transition. I think you're right.
We got the housing crisis, the whole, Trump thing. Now we got the COVID. Now we're
going to now everybody's starting to be worried about nuclear war. It's like
wages have been, real wages are falling. Cost of living is going up. Yeah, it's tough.
And so it's very tough to be able to say, like, how much of this is just a return to a slightly lower baseline because you've recently been really sick, and that's layered over everything. We'll get to that kind of a little bit more later. I don't know that we need to...
It's worth mentioning, though, fatigue's by far and away the biggest symptom of this long COVID stuff.
Yeah.
That's what we're talking about mostly here.
Right. But there's also these abnormalities in actual lab tests, right? So you have elevated white blood cells, you have a lower amount of red blood cells, your fat production isn't normal, whether that's the way that you're packaging cholesterol or if it's the way that you're producing your lipids, or you have the wrong kind of lipids in your blood.
Could affect hormones.
Yeah, like your liver function's not ideal. You're getting a lot of these microclots. There are definitely
micro dysregulation.
Yeah, so it's just it's this kind of disaster in the aftermath. And we don't have a lot to compare it to. And this is the worst part, because in the text, we'll get to this, but they're like, look, people really don't recover after they go to the hospital. They have this extended kind of fugue state of illness that extends after going to the hospital. So if that's the case that's happening here, it would be very difficult for us to tell for another year or so, if you come back to these people, will they be okay? And maybe they will, maybe they won't. Time will tell. It's very, very, very ambiguous right now. Yeah, so there's just basically all kinds of things. So you have these neurodegenerative disorders such as Parkinson's, cognitive impairment from brain fog to accelerations of Alzheimer's disease that are described in patients who have had acute COVID. Also, a range of psychiatric disorders, like we've talked about all of this, but it's basically that you look at somebody who's been sick, or maybe not sick, which is jury's still out on whether or not you can say that, and you start to see all of these accelerated problems. But Alzheimer's is also accelerated by being deeply socially isolated. And if you've had COVID and you've been sick for a long time, then the two would layer onto each other. And there's very little effort to tease the two apart, because I think people want to be able to identify what long COVID is, because they do want to help people. And so they don't want to be like the medical community has been with chronic fatigue syndrome and just be like, oh, it's all in your head. And so you really have to balance these two as you're going through the data to see what the complete picture is.
Yeah, and it's I said this already, I think earlier before we started the lecture, but it's not just because it's in your head doesn't mean it's not physiologically important too, or that it's as easy as just changing your mind. It's not quite so simple.
I mean, so there's a really, really interesting book on psychosomatic illness. It's called It's All in Your Head by this woman named Suzanne O'Sullivan. And Suzanne O'Sullivan was working as a neuropsychiatric resident in the National Health Services in Britain. And she was working at a clinic where they were dealing with treatment-resistant epileptics. And what she discovered is that some 30% of the patients that she was seeing.
These are people who have seizures, by the way.
Yeah, so they have seizures. They would do lab tests on them, and they would discover that they don't actually have epilepsy because they don't have the accompanying electrochemical disruptions of the nervous system. Like
when you have an epileptic seizure, it's like unplugging the computer. Like you have a complete synchronous signal from many regions of the brain all at once.
And so you actually can look at this under EKG, and so you can see the waves basically bounce around in the brain. And so a lot of these treatment-resistant epileptics are people that were having seizures. They were epileptic on the surface.
Symptomatically.
But when you did the lab tests, you found that they weren't. And so for me, And
this was really hard on the patients.
It was very hard on the patients.
I mean, the last thing they want to be told is that you don't actually have this disease that you think you have.
And she was very good at getting them into counseling and therapy and finding, the book is told through a series of anecdotes where she talks about all of these different people's stories. The point that I want to make here, though, is that when you start to see lab manifestations of like heart inflammation, it's harder for me to say that it's a psychosomatic illness because I don't have the clear mechanism for how your mindset could lead to...
Really? Even with like the hypothalamic pituitary axis and all this stuff. I mean, I just think that we're so young at understanding how the brain influences the immune system. Like when I was in college, it was not a thing. Like I was a neuroscience major in college. This was like 100 years ago. But it occurred to me that the immune system was very similar, like in that they're chemically signaling cells that talk to one another. And they were clearly interacting with the hypothalamus, but nobody was studying it. And they're just now starting to study it, and it's crazy what they're finding out. Like there's cells that are half nerve, half immune cells, right? These Philippodia cells or something like that in the gut. It's not this ambiguous thing. Like the hormone structures are being affected which by your experience on Earth, the world that you're in, psychologically, spiritually, however you want to put it. And that is literally affecting the hormone structure, the hormone output, which is either, obviously, what's the best treatment for COVID, for putting down a massive inflammation response? Steroids, hormones, right? Those are the same sort of hormones of the same class that they're called corticosteroids because they mimic the ones that are emitted from the cortex of your adrenal glands, which it's a hormone response. So it's not clear to me that just because we don't know exactly how that works, that it's not very physical and very much a real thing.
I think that maybe where my hesitation lies is not that I don't believe that it can't be a real thing. It's more that it's hard for me to grapple with the idea that it would be so that the responsibility then falls on the individual person.
I don't think that's the case at all.
That's what I worry, right? Because when you start telling people like, hey, this might have to do with the fact that you have problems with the way that the world is going and you're very stressed out and you're unhappy, then for me, what I fear is that the next level is the cult of health and busyness comes through and is just like, well, what you need to do is you need to take care of all of this by yourself because you're the one who's sick. And if you fixed yourself, you wouldn't have these things. And I'm like, I worry that that's the runaway train. You know what I mean?
Yeah, I mean, that's the rough thing. Like realizing that you're anxious or whatever is one thing. Changing it is nearly impossible. You know, it can be done. There's things you can do, obviously. I mean, it's not impossible, but it's very, very difficult.
Well, and especially if what you're trying to do is you're trying to change...
Physiologically anxious, you know?
But you could be physiologically anxious because the society in which you live is pressing upon you from so many directions that there's no other way for you to be. And so without changing the society, can you really fix things for people? And that's...
there's things you can do, right, to manage your stress.
But that's like, so I feel this way about like meditation sometimes, right? Where people are like, you should meditate. And I'm like, not if what you should do is you should get to work on fixing the problems that you already know are there, right? So it's like, does telling people to find ways around their anxiety about the system lead those systems to be extended ad nauseum? My gut feeling is that yes, it does.
But yeah.
And it's like what people need is people need community health, they need clean water, they need good housing, they need education, they need all of these services. They don't. And so there's this fear that I have where the more that we start talking about, hey, this is internal, especially for things like the immune system, the less we talk about how much systemic change still has to happen in order to make this a livable world. And so
yeah, there's a huge confusion between it, whether it's internal and whether it's real too. I see this get confused all the time. The word real is really hard for people to handle because they feel like you're telling them it's not real if you're telling them that they have psychiatric symptoms or whatever as a result, and they want something more hard concrete than that, right?
Perhaps. I mean,
certainly exercise, meditation can help with inflammation. But
it can.
Yeah.
But it's also just like if people are working crummy jobs and they're really stressed out and they have long commutes and they're very poor and they're very uncertain, it's like meditation's not going to solve those problems.
For sure.
It might make you tolerant of those problems for long enough to be able to continue and not be sick. But there's also this thing to me where I feel like there is a great danger to putting it on individual health, rather than focusing on the idea that perhaps people are sick because the society, the structures in which they operate are sick. Do you know what I mean? Like,
and that's cool. I mean, I think the fact that they're categorizing all these symptoms and these clusters of symptoms allows other people to sit back and be like, what's going on here? You know, is this because the virus itself is pernicious, or is it because of the way that our world is set up too? And it's probably a little bit of both. But certainly, if we learned anything from COVID, a lot of it has to do with how it's all this pathology is happening at the interface between us as human beings and this external, bag of genes and protein, right?
Yeah, it's throwing everything into just chaos.
There's something that both sides of that story are telling an equally important part.
Mm-hmm. So yeah, I mean, I guess to wrap that up before we move on to the further sequelae is, I think that when you talk about the immune system and you talk about dysfunction of the immune system, there's going to be some cluster of people that have genetic mutations. And that cluster of people probably will not be helped by meditation, and it's not psychosomatic, and they have some kind of autoimmunity. Then you have another cluster of people who I think is probably larger, and that cluster of people is at the intersection between immunity and stress and society and their own life experience and their own internal world. And I don't have the language to talk about that without stepping on toes. I feel like I really don't want anyone to walk away with the feeling that it's their responsibility to exclusively their responsibility to fix this, because there are much larger things at work that could lead to something like post-COVID or long COVID. That
we all nobody's starting from the same place anyways, even physically, right? None of us had the same upbringing, as the same genes. We're just also very different.
Exactly.
But there are some things you might be able to do.
Exactly. I mean, and the list of things that are pretty intense, they are related to the immune system for the large part, right? Myocardial inflammation in 60% of patients, independent of pre-existing conditions. Even athletes are getting myocarditis, 15% of these very, very small group of athletes. There's like weird morphological changes in about a third of people after hospitalization, which now this seems to suggest that we're talking about a pretty severe COVID infection, because you're not getting hospitalized if you're not getting something pretty severe. Let's see what else.
It didn't make it into either one of these papers, but I just read here that there was a study that just came out in the Lancet that showed that the vaccine did actually improve odds with long COVID for whatever reason.
2 doses, it says, yeah?
Yeah.
So it's worth of either one or of...
No, I'll pull up the paper.
So, we've got all of these gastrointestinal symptoms that we've talked about: loss of appetite, abdominal pain, nausea, weight loss, weird bowel patterns, irritable bowel syndrome. So there's a remodeling that's happening after being exposed to SARS-CoV-2. And there's also this prolonged fecal shedding, which suggests that the virus is surviving in the intestines for a long period of time, whether that's because it's reproducing on the cells that line the mucosa, which to me makes sense because they're so frequently updated that you're constantly having new cells to show up to get infected that might not have worked up their sort of cellular immune pathways yet. This is also really fascinating. So owing to the expression of ACE2 by pancreatic islet cells, the SARS coronavirus was found to cause islet cell injury and result in acute type 1 diabetes mellitus. What that means is that if you can't produce insulin, these islet cells in the pancreas are responsible for this. The virus itself can destroy those cells, and when those cells are destroyed, you become functionally diabetic because they don't regrow. And this actually happened to one of Dr. DeLay's colleagues a long time ago.
Yeah, it's terrible. He was in med school. He just became diabetic overnight. He got a really bad flu, cold type thing, and his pancreas died.
His insulin cells died. The pancreas is larger than that.
Right. The skeleton of the pancreas didn't die. Fair enough. Yeah, it was really bad. And I had never heard of anything like that at that time. I started thinking about getting the flu a little bit differently.
Right. So these are things that happen. It's hard to argue with. There's, we're at this entire portion of the paper is kind of looking at symptoms. Where did you want to go? Did you want to go to the figure? Let's look at this figure.
Sure. The other paper is going to repeat some of this, so we don't have to nail everything right away.
Exactly.
We can jump over to the other paper. I like the figures from the other paper better.
So they're basically saying that at the center of this figure, let's zoom out a little bit. Not that far. So
it's basically 3 clusters of causative. I don't know. I guess they're getting into mechanism to some extent, right?
Sure.
They're like, there's 3 things that could be going on here. 1, your body fails to recover its inflammatory response. 2, there's autoimmunity being developed. So there's other parts, the body's starting to attack itself, essentially, because it realizes that there's cells that are infected it hasn't got dealt with yet, or it believes so. It's highly sensitive. Or the virus is literally still living inside of you somehow or another, which will maybe leads to the third paper. Interestingly, I think the one that's, the data, the one that's most interesting, I won't say that. The one that has the most concrete evidence towards it seems to be the persistent inflammation, because they're able to track altered cytokine productions in these patients at long range. However, it seems like the data on that is rather conflicting. If we go to the body of the paper, or maybe it's in the other paper, there's studies that argue about whether these different factors go up or down depending on who they look at. But it makes sense that if you have somehow exhausted your immune system, particularly the compartment like the regulatory T cells, the compartment that's supposed to shut everything off, if you've stressed all of this, it's possible that it's not able to do its job, and you basically just don't turn off your fire hoses at the end of the day.
And all of this leads as a funnel to this post-COVID-19 syndrome, right? So there's a bunch of things that viral persistence, persistent inflammation, and autoimmunity cause, which is fibrosis and scarring, cardiac remodeling, neural inflammation and remodeling, and dysautonomia. What is dysautonomia? I always forget.
Where is it?
On the figure?
I'm looking at the text right now. All of those are the direct result of those cytokines too, which supports the idea that if they're detectably up, it could trace, right? Because you have a cluster of symptoms and you have a cluster of biomarkers, and they correlate together. It makes it a lot more convincing.
Yeah. Dysautonomia is the involuntary functioning of your nervous system. And so your blood pressure and your heart rate and your sweating patterns and responses get to be kind of screwy in the aftermath of all of this. And again, the things that lead to it, for the most part, are this persistent inflammation, because right now they have it drawn where viral persistence and persistent inflammation and autoimmunity are kind of all on the same level. But
what they're saying is they've seen this in other diseases too. Like they've seen cases before where there's cardiac remodeling, you have increased IL-6, IL-1B, and TNF. Or the same for neuroinflammation, dysrhythmias, renal injury, all this stuff.
Where are you reading this?
From the text. Each one has its own citation for how those immune markers are involved.
Sure, where are you in the text?
Oh, sorry, page 198, right there. You highlighted it too.
Right, okay. Yeah, so it's these cytokines are so powerful in the body that when you're bathed in them for a long period of time, it's crazy to expect that you'd be able to come out on the other side and not have some kind of dysregulation that takes time to put back together. IL-6 especially is it's responsible for opening up capillaries and opening up passages in between cells, and so it makes a lot of sense that in the aftermath of IL-6, you're going to have a lot of dysregulation and dysfunction as things start to kind of return back to baseline.
And I think the back to baseline program is what's really at the center of the discussion for the back half of this paper, if I recall correctly.
Mm-hmm.
And we're really focusing on this idea that there is such a thing as immune homeostasis in the first place, where your immune system is just totally at ease, and that it's very difficult to return to following a severe illness like this. And so separating out the concept of long COVID from the return to immune homeostasis following extreme illness is a very difficult task that the authors acknowledge going through here.
Right. And I mean, they talk a little bit about the various ways in which that it can be detected. They do make the point that no direct evidence was available from the present study when they're talking about potential ways that immune system cells, these effector T and B cells, which are the ones that are actually encountering the antigen responding to it, like they don't really know if there's a mechanistic link, because it's not enough for a paper to be able to show that, hey, these guys are elevated at the same time that we're seeing these persistent problems. You actually have to have a mechanistic sort of logical line that you can demonstrate of cause and effect in order to be able to say that this is what is causing long COVID. And it's really fascinating because I'm repeating myself, but a year ago, this is exactly what we were looking at when we were looking at COVID itself. There was just this huge amount of kind of just open palms and shrugging of just, well, we don't know what's causing it, and hopefully we'll find out eventually.
What's really interesting about the B and T cell populations expanding is later in the paper they mentioned that the B cell pool continues to clonally develop even after 6 months from following the infection. And the antibodies are different. They have more hypermutation. And
do you know where
sorry,
you are?
I'm on page, I'm about midway through page 199.
I see.
Second paragraph down here. Reports of, yeah, you highlighted it too. So that's what's really fascinating, is that the body is, at least in some of these patients, I'm not sure what the prevalence of this is, they're still acting like they have a virus, in support of the idea that this is specific to COVID and not just specific to inflammatory disease in general. It seems like the body in some, I don't know what percentage of the cases here.
So a memory B cell, so when it says memory B cell continues to display clonal turnover, can you?
Yeah.
We haven't done too much.
Yeah, it's been a while
immunology.
anyways, since one of the first couple lectures. So remember, when you're in the process of making antibodies, basically you're trying to crack a combination lock. It's kind of like Bitcoin or something. And you're just trying out all of these combinations and trying them and trying them and trying them. And ideally, once you crack the code, you just crank out a bunch of those antibody producing cells that have the perfect code. But that, at least according to this, this statement in the paper in the middle here, which is referenced twice, we can follow up on that. But at least according to this statement, they're saying that the key generator is still running in the background for up to 6 months after the infection. Now, is that because the virus itself is mutating and they're confounding the production of new antibodies for the original, or is it because the virus is truly persisting inside of the person in some way or another? It's not clear at all, but it's interesting. It for me, it lends credibility to the argument that this is a COVID related, at least in some portion of this is a COVID-related constellation and not a generalized situation.
And this goes back to the idea about the epilepsy or the psychosomatic epilepsy, right? Where one thing that they couldn't identify in these patients that made it obvious that it wasn't epilepsy was this absence of electrical storms. If you could never discover any indication that the pool of COVID-specific B cells was still developing and expanding and responding to some kind of epitope, then it would be much harder to make the case that there's something to do with resident remnant virus. But since you do have this evidence, if it bears out, that's a clue in the direction of, okay, there is some factor of it. But if it's very hard to find, right, you have to look at levels. Like how much of your virus is there? Like, are there 2 memory B cells that have expanded over the time of this, or is it?
And is it happening in how many patients is it happening in? Yeah, there's so many questions.
Yeah.
Have they been exposed to other variants? Like,
Yeah.
it's very hard. It is interesting, but it's too hard to make a conclusion off what it means right there.
Mm-hmm.
All right, so then they talk about some of the other organ systems.
Where are you? Sorry.
Sorry, I was just keeping going right there. You want to go back?
Aren't I exactly where we were?
Mhm. Here?
Mm-hmm.
Oh,
Where?
we skipped a bunch.
Sorry, tell me. I'm not sure where you are. Go ahead.
Maybe we've already mentioned the different organ systems sufficiently. Maybe we can just jump over to the other paper.
I want to before we go, which I think that's a good idea. But before we go, I want to touch a little bit on their conclusions, which they talk about here with high dimensional profiling of the systemic and local immunity of patients with COVID-19. And they're basically saying what I feel like I've been saying ever since I started looking at COVID, which is that you need a lot more information about individuals before, during, and after illness in order to be able to say something that's robust. There's so much interpersonal variation that the baseline that you have to be comparing people to is not some generalized baseline of what it means to be healthy, unhealthy, sick, not sick, elevated cytokines or depressed cytokines. You have to have a profile of each person that has been done carefully enough to be able to say, is it our looking that has made us notice these things? Has this person always been this way? Or has this really and truly been exacerbated by what they went through? And the only way to do that is to have really good controls, where here it says additional controls would include patients with other viral infections and patients with autoinflammatory diseases.
I forgot to mention that too. They say on the other side of this paper that other viruses do very similar things. Where is it? Here we go. The induction of autoimmunity after a number of viral infections, including parvovirus, blah 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 blah. So they've seen this and they've seen similar constellations in other viral infections.
Exactly. So you're basically in a condition where you know that there's other viruses that do this, but you don't have the ability to say if COVID is special or not because you've never had the chance to look. And so they're basically insisting on the fact that in order to have robust conclusions going forward in the future, you're going to have you need to have a lot more medical details about specific people in order
across
to
time. Yeah, it would be
time.
beautiful if everybody had their whole seriological history. If that's why you guys remember the Theranos nightmare. It was there was this whole debacle where this company was promoting this really tiny, small volume blood draws. You could just go into like Walgreens or something and get a blood stick and find out all of these different things about
a finger stick. They'd take just a drop of blood.
Just a little drop of blood. Very harmless. It's something you could do very cheap. You could go and order whatever blood tests you want. You don't need a doctor. Very cool stuff. It turned out to be a lie, but it's a great idea. And the future is gonna, if we really want to be able to get a grip on these sorts of things as they emerge, we're gonna have to know our own histories, and very detailed molecular histories about ourselves, I think, too.
And that's gonna be hard.
That's gonna be hard, but we'll get there.
All right, so let's take a look. The other paper has some other interesting aspects, and then we can get to the transposons. Let's try to go. They say a lot of the same things.
They do. I think the mechanistic stuff might be a little more interesting in this paper. Their figure is really cool.
Yes, let's look at the figures for this one.
At least seeing it twice too will help you get stuck in your head a little bit.
All right.
Which is good. So.
This is the same figure that we've seen pretty much everywhere, which is just these are the various systems that have ACE2 receptors. And these are the pathological sequelae that each one of these organ systems has. Every single figure, I think that every single paper by scientific convention has to have this same collection of information. Personally, I think that it would be better for papers to use the space for something else, but so it goes. these are all, so this is a huge meta-analysis. Meta-analysis is basically going and looking at specific papers that have done some kind of either observational study, or they have done an experimental study, or they have collected data in some way that these people who are writing this meta-analysis think is good. And they assemble all of those papers, and through the application of complex statistics are able to say that in aggregate, these are the effects that we appear to be seeing in the literature. If you remember, we were looking at the website for the various treatments. That was meta-analysis.
I think that it says here they included 218 different studies at the end of the day.
And you can see that these are not all very high-powered studies. 140 people, 100 people. There's 58 people. There's some of these that are larger, right? You have 1,000, 2,000. I guess this is almost 2,000.
And they're all done in different ways in different countries at different different hospitals, and with different markers being collected, and various, it's not super clean, but it's the best you can do.
And it's like,
honestly, this whole thing just seems like a lesson for the future. Like, how would this go better in the future? How could we standardize the way that we collect and share data as a globe, at least, the cooperating countries? How could we do it if we wanted to? What would that look like? Because it'd almost be like reverse engineering one of these meta studies to actually be meaningful could help us design a better scientific, epidemiological research base in the future.
Because the problem with this is garbage in, garbage out. If you do a meta-analysis and every single study that you're looking at is underpowered, done in a way that is observational and is collecting their data in kind of shaky ways, your meta-analysis isn't gonna...
Like, if I told you guys we're gonna go out into Portland and find out what the average height of people is, but somebody just goes to a preschool or something and measures all the people. Somebody forgets to tell people to take their shoes off. Some people are measuring in millimeters. Some people are measuring in centimeters. Some people are just measuring in hands or something. It gets impossible to actually say anything. We can maybe make some rough statements, but we won't have any sort of scientific precision at the end of the day. So, maybe one of you guys will go into epidemiology in the future and get to, at some point in your lifetime, to serve on one of these international committees that's talking about how we can change the way that all these nations deposit their data and keep track of it and standardize it. And that's the painful way that it's going to have to change in the future, I'm afraid. Cool. This is the figure I liked.
Yeah, you want to walk us through it?
Yeah. So most of these things we've talked about. It's just, I think it's important to see the picture. So one of the more solid pieces of evidence for persistent long COVID is this: when they look at people's chest x-rays, they see this ground glass presentation deep into the future after your infection, which makes sense because I still have scars on my body that I got when I was a child. Once your body's gone into this scarring process, which is essentially, if I could be liberal and say that this, what do they call it?
Fibronectin?
No, it's like this process of making fibers. Yeah, fibrosis. That's it. This fibrosis is very much a scarring process where it's trying to keep the structural integrity of the lung in place in areas where it's been injured. And so they know that involves different immune signalings that we've identified from the different studies already. IL-6, IL-1 beta, so forth, which are elicited by when the tissue is immediately damaged, both by the immune system in an autoimmune fashion. T cells come in and try to kill these infected cells. Okay, that's fine, except now you've killed off the important parts of your lung that are doing the gas exchange, so that's not good. But when your body comes in to repair it, it's doing a sort of bang-up job, it's patching things up. It's not actually planning for the future. So, right now, we just bought a new house, and our house, when we bought it, unbeknownst to us, there was a nice hole in the roof right over the kitchen. one day we were just sitting working at the computers and like a waterfall started. And so what do we do? Well, it's pouring outside, so we patched it up really quick with tar, basically. Now we have a bunch of tar on the roof, but is that something that's going to work out for us in the long run? Probably not. In fact, we've actually made a mess out of our roof, and we're going to have to replace that whole area now, if not the entire roof. And honestly, if we just replaced the whole roof in the first place, it might have worked out better for us in the long run. But your body can't do that. It can't just replace all these cells very quickly, right? It needs that lung not to be leaking gas, leaking blood, things like that. So it's going to plug the holes.
There's some very interesting research on scar tissue and the sort of these fibrous processes, which suggests that fasting in the aftermath of an event like this can help clear scar tissue, where what happens is that when the body starts to need sugars, or it starts to need protein, or it starts to need fuel, basically, I guess fat doesn't work for this, but if you fast for, 24, 48, 72 hours once a month or something, or after a long illness once you've fully recovered, that kind of periodic burst of what's called autophagy can help the body activate the resident macrophages that are able to basically go around, collect all of these things, and then put the raw materials into the blood, because eventually that's where everything goes. And then it gets the things that are actually used by the body can be devoured by the immune system to remove all of these plaques and to remove all of these sorts of things, and then to pass them along to the cells that are using them. I've heard this to be really effective against acne. I've heard this effective against scars. I've heard this effective against cysts, where if you induce the body to go through a period of self-cannibalization, you can recover some of this. And it would be really interesting to see, after somebody's gone through this kind of immune catastrophe, if fasting for some controlled period of time could actually help. I haven't seen any papers of that, so if anybody does, I'm particularly interested in that, because it seems like such a small and yet powerful change, because we already know that this kind of autophagy can help decrease the incidence rate of cancer. It seems to contribute to longevity, and it seems like it might help with long COVID.
It's also really hard to do if you're a hungry person like me. I love the idea of fasting, but I just always fall short of it.
I've only ever fasted for a long time once, and I nearly crashed my car. Do you remember that?
Yes.
Don't drive and fast. We went somewhere, and then we got back in the car, and for some reason I was driving, and I just backed straight into the person I was parallel parked in front of. You were so upset with me.
I want to hear other people's stories of fasting later. It's a really interesting idea. It's one of those things that there's always a community willing to stick up for it.
Definitely.
There's also the Biosphere guy who it didn't really work out for. So I don't know.
Yeah, there was this project in the 80s called Biosphere 2, where there was a bunch of people who were really interested in the idea of ecological sustainability and taking care of the planet. And what they wanted to do is they wanted to do an experience that would demonstrate to people how important it was to take care of the planet. And so they built this huge dome structure in the desert of Tucson, Arizona, and they seeded it with the life from all 6 biomes on Earth. So they had a forest, they had an ocean, they had a desert, they had a prairie, they had I don't think they had a tundra. But they basically then locked themselves into it, and it was perfectly sealed, so oxygen couldn't get in and out. And they closed themselves into it for 2 years. And over the course of 2 years, they basically farmed and starved because they ran out of food. They started to run out of food at some point, and so they went on this super hyper calorie restrictive diet. And one of the physicians, the main guy who was there, he was this huge proponent of fasting, because there is an entire movement that believes that the only thing that will extend your lifespan is living eternally calorie restricted. And there is some reason to believe that is the case, because if you take experimental mice, experimental animals, I think rats as well, and you maintain them on this very, very minimalist diet where they're always in a calorie deficit, or they're right at their needs, they're not necessarily losing any weight, but they're burning absolutely everything they eat, and you balance the diet properly, it's one of the only things that has been consistently shown to increase lifespan. And one of the reasons that they think is because it triggers this autophagy.
But it didn't work out for their main doctor, I guess.
Their main doctor ended up dying really young. It's a fascinating story. I think that there is a Netflix documentary about this, and I think it's called like the Biosphereans or Biosphere 2. It came out just like maybe a year ago.
It's a really fun story for sure. It also it's still there, right?
Yeah, it's still there. You can go visit it. If you're ever in Arizona, go check out Biosphere 2. It's owned by Steve Bannon.
I was gonna say, I thought some
which is like one of the weirdest.
Columbia owned it for a little while.
Yeah, Columbia owned it and then they sold it to Steve Bannon.
The whole story is fascinating. Anyways.
Back to the cardiomyocytes. Walk us through.
This is the same story as with the lobular cells.
Except we're talking here about cardiomyocytes. So the reason that you have the same development of fibrosis in the heart lungs, but you also have this displacement of desmosomal proteins. And this displacement of desmosomal proteins is you're literally breaking the way that the cells hold to each other inside of the heart. So in the last paper, we talked about cardiac remodeling and ventricular abnormalities. And this is basically why, because you're breaking apart the structures of how the cells hold to each other. And so that the heart is under a great deal of pressure. It exerts a great deal of force, and that's pushing it into these strange conformations, and then you get locked into those conformations because you start to produce these fibromyoblasts that your cells are like, well, we need to do something to support the structure that's been broken down. So we replace these desmosomal proteins that are on the surface of cells with dedicated structural cells. And that's what's causing this long-term sort of fibrotic persistence. And then obviously you also have this huge inflammatory response and all the death of things.
And then they're also bringing in the fact that if you have autonomic dysfunction, the heart is controlled by that system to some extent, right? If you get very anxious, your heart goes nuts. This is an autonomic response. So if you have dysregulation or imbalance in your autonomic function, that's going to contribute to poor cardiac function. I mean, the heart's an amazing symphony of firing, right? All those cells have to be in concert, creating these waves of motion throughout them as they contract. So it's no small task for your nervous system to maintain.
Definitely. So finally we have, or I guess not finally, second to last, we have these neuropsychiatric sequelae. And so they're kind of here demonstrating all of the different ways in which these things fold onto each other. And so...
yeah, a lot of this comes down to inflammatory damage and the activation of these microglia that are, continuing to provide this stimulation long beyond when necessary. Self-perpetuating neurotoxicity, activation of pro-inflammatory microglia. Right,
so microglia are the brain's immune cells. And so when you activate these immune cells, what they're going to do is they're going to perpetuate an immune response. And so you have negative impacts on the axonal cells, the ones that are actually sending signals. And so your brain fog might be because you're actually seeing a decrease in the cell number in the brain. Like I think Lily mentioned this a while ago, or somebody else brought a paper about decreases in gray matter versus white matter, where it was like listed that there was specific abnormalities in gray matter. And microglial neurotoxicity is going to be the mechanism by which that you start to lose these brain cells, basically. We never did get to the bottom of if it's equal in white or gray. But you then have so the brain is normally not surveilled by the immune system. And this is very, very important because it's not surveilled by the body's immune system, right? So there's a blood-brain barrier, and it's developed in such a way where these immune cells that are capable of doing damage are not able to get across the blood-brain barrier, because if they did and they encountered inflammation, then they would start to destroy things. And in the case of severe COVID and in the aftermath of COVID, you have leukocyte infiltration, which means that this normal barrier that's there to prevent these sort of bomber cells from getting in is broken down, once they get into the blood, into the brain, then they can also start to destroy cells. So you have your brain's immune system, these microglia that are producing inflammatory cytokines that are killing cells. Then you have normal innate immune cells that are coming in and they're destroying brain cells. You also have blood clots, where the blood clots are basically stopping the flow of blood, and so you have these micro strokes. And again, we're talking about a relatively infrequent condition, because we're not talking about everybody who has COVID. We're not talking about every single person. But in the cases where you do have long COVID, these are the proposed mechanisms.
And then, of course, all of those are compounded by any sort of muscle damage that's happening as well, which is the fourth panel.
Right. So it's like you can have physical damage to the muscle. They look and they say that you can find direct damage to the cells.
Like if you work out really hard, you actually do feel tired kind of in the muscularly.
And if you have a virus that's destroyed a lot of your muscle cells, you're also going to feel tired. That makes sense. There's also some indication.
Due to fiber damage too, right?
Yeah, exactly.
They're saying there's actual damage to the fibers.
Right. And so all of these together sum to create this kind of this tiredness. You hear people expressing these psychological and social factors a lot of, I'm so tired, I'm so over it, I hate this. But all of these things together physically contributes to creating the persistent fatigue state. This is this just their meta-analysis framework?
It's like it's how they're gonna go about. They recommend surveilling it and treating it.
Oh, this is the nice rapid guidelines. So there's basically there's an organization that is in charge of coming together and proposing ways of treating long-term disease, right? And so they start with a COVID-19 diagnosis. You go through the entire thing, and then you identify long COVID, and it's this whole cascade of events that happen afterwards, where you then have to start monitoring and taking care and planning these people's care, finding ways to manage their care, because if this ends up being a long-term persistent problem, we're going to have a relatively large population of people that have
large infirm population.
Yeah, we're going to have a large infirm population that sticks around for a long time. And so organizations are basically trying to figure out how to deal with this. But, management, it's like give advice and information on self-management. Support people in discussing returning to work /education, but it's not like, support people by helping them find a job that works with their new current state. It's like this feels very much like 2 years ago at this point, I guess, where people really didn't know what was happening and how they were going to deal with this. And so we saw a lot of charts like this where they're like, well, these are all of the different things that we could do. NICE actually got into a lot of trouble
Trying to find out who they are.
With people who actually have chronic fatigue syndrome, right? So it's like basically
it's a division of the NHS, right? So it's Britain's CDC, basically.
I think so, yeah. So they have they use their chronic fatigue syndrome guidelines for suggesting how to treat COVID patients, and they're like, well, you can get cognitive behavioral therapy and graded exercise therapy, which is basically like get therapy and walk around. But coming from a public health organization, of the directive of, well, just walk it off doesn't seem like a particularly caring or effective directive. But I think that this has less to do with the fact that they don't care and more to do with the fact that they just don't really know what to do. Because we're still at the point where we're trying to figure out if this is even a real, like how to define this, and where does it begin and where does it end. That's what I mean by that.
Seems like NICE is also a budgeting, like they are a budget advisory institution as well, so they decide what money gets spent on what. And so a lot of this is aimed at how do we do this in the most cost-effective manner, and they've been criticized for this in the past,
Mm-hmm.
not really being objective. So
Yeah, I mean, they also are basically like talking about just offering people various drugs that would help remove brain fog, which also feels unsatisfying, right? Like modafinil is this neuromodulator that they give in the military to make pilots not fall asleep when they're on long missions.
do you guys ever see Limitless, the movie?
Yeah, modafinil is basically the drug that movie was based off of. And so they're like, well, we'll just give drugs to people and it will fix them. And it's like, maybe it will, but it seems like you can't really figure out one way or another what is actually going to be good for people.
I think the guy starts like murdering people in his sleep at some point in that movie. It works though. It does help him concentrate.
And then they start to just go down the list of repurposing drugs, which is antihistamines, hyperbaric oxygen, emerging treatments. They're trying to think about antidepressants. Antidepressants and antihistamines often have an anti-inflammatory effect. So it makes sense that you would think of giving them to an inflamed condition.
Beta blockers even.
Yeah, basically anything that kind of just tamps down the response in order to get the body to just be comfortable with the fact that there's this new presence in it. It kind of makes me think, though, of like anti-rejection medications, where if you have something that stays in your body for a long time and now it's just part of your body, but your body never gets used to the fact that it's there, do you have to be on a lifelong treatment plan for it?
For like organ donations?
Well, for organ donors, definitely, but I'm thinking about for something like a virus.
Yeah, long term. Yeah, it's hard. Or I mean, yeah, maybe.
Right? It's not
clear either that you wouldn't want some of those signaling cascades to work for other diseases you might encounter, like cancer or other infectious diseases that might rival COVID, that we can't even imagine right now, or that we can imagine, like tuberculosis or something.
I mean, I think that the wrap up here is basically that we don't yet know a lot of very specific things about long COVID. And a lot of the facts that we don't know have to do with, do all viruses do this? Is this just the arc that you travel after you've been really sick? Or is this something that is wholly unique and specific to COVID? And so if you follow this for another year, I bet you that we're going to start to see much clearer results.
Yeah, we told you it was going to be a disappointing conclusion-wise. It's going to be a disappointing lecture.
I mean, I personally think that we're going to find out that this dissipates over a long period of time, because that's my sense with other severe illnesses too. You see somebody who has just finished chemotherapy, and they're pretty messed up. You see somebody 5 years after they finished chemotherapy, and they're pretty good.
You see a little kid that 5 years ago had chemotherapy looked great.
Right. So I think that we're really starting to look closely at the fact that something bad happens to your body after severe illness, but that's to be expected. The question is, how long will it take to resolve? If I think that making predictions and then being wrong or right about them is worthwhile. Like, keep a systematic record of the predictions you make so that you actually can tell if you evaluate things correctly. So I'm going on record right here, right now. I think that long COVID is probably about a year long, maybe a little longer, but I do think that people return to baseline.
It's optimistic.
It is very optimistic.
I wonder if people will still be studying if the money will be there to study long COVID after more than a year too. So that's an interesting question.
That's not even.
I don't know. We'll keep throwing these lectures up as we teach them over the years. If anybody even still wants to take immunology of COVID after, there's more exciting things, and it'll be interesting to look back in a few years.
So let's do this one real quick.
Real quick. So this the final paper was just kind of thrown in there because these authors make a really interesting point about the potential for some of these lasting PCR tests and maybe some of the symptom clusters to be a result of persistent virus that's actually been incorporated into your genes.
Pieces of virus, not the entire variant.
Thank you. Yeah, so it really only bears mentioning because it's really at the edge of what is known. This paper has been criticized a lot. It's worth mentioning that. because people don't like it, not because of the way they did it or anything. This is actually in PNAS, which is a fantastic journal. It's the Proceedings of the National Academy, which is sort of our Camelot of science here in the United States. But nonetheless, this is a very technical paper, so I didn't really want to go into the details of how they did all of these studies. What they've basically tried to present is, this is all done in vitro in a cell line, cell lines. They've basically shown that there is a potential mechanism by which these transposable elements in your own genome that represent some incredible amount of your own genome, like 20%.
It actually says...
Yeah, believe it. In this paper, they say it's like 21%.
17%.
17%. Okay. Different sources say different things. That's it. That's crazy. So you want to tell them about what transposable elements are a little bit?
All right.
Wait, hold on one second. Let me just sum up the paper. Basically, the paper is saying that, yes, the SARS-CoV-2 is an RNA virus. That should be a very temporary thing because it doesn't even get involved in what's going on in the nucleus. But in some rare cases, it seems like it's at least possible that it can use your own reverse transcription mechanisms, particularly this one with the retrotransposase, to insert the virus into your DNA. So let's rewind that a second.
So transposons are called jumping
I have
genes.
some slides if you want them or not.
You want to pull them up?
Sure.
All right. Did you have them open already?
No,
All
it's
right.
okay. They're not that good. Just talk.
All right. So these transposons in general are elements that can move freely through the DNA. And what that means is that you have enzymes in your body that are able to take these elements and, because of their genomic architecture, cut them out and then put them somewhere else. You need to have a protein to do this. It's called a transposase, and your transposon has to have some specific like repeats on it. Where basically what you see here is that there's a sequence that is just this AATTTT. So anywhere in the genome where you have this sequence, it's possible for your template, which is represented here as this red line, could jump onto this sequence. Like once you have the transposase, which cuts at this sequence, and it's holding the transposon, and then it can just basically clip the transposon into this place, and the transposon then is able to, because of where you cut this, integrate into the other strand of DNA, because you cut it at this AATTT, and so on this side you have the transposon is able to bind to the TTTs, and then on this side it binds to the AAA, and then it basically is snug and perfectly at home. Normally, they're not moving around that much because your endogenous levels of transposase are pretty low. However, it's possible to have an increase in your levels of transposase, and when that happens, you can have these jumping genes become destabilized and start to move around the genome and end up in various places. And there's a lot of different sites where these transposable elements can jump to, and there's some indication that activation of them is actually leading to the super large number of cancer cases that we have in our population today, where cancer cases, especially sort of colon cancers and these various environmental cancers, are going up. Lots of other ones are going down, don't get me wrong. Like lung cancer is going down. We're seeing a lot of these gastrointestinal ones though rise. And that might be because of the fact that there's something either in the food supply or in the amount of stress that people are having where it's activating transposable elements in those organ systems, which are then causing cancer. Because you can imagine cancer is often a disease of cell growth regulation. You mutate a checkpoint that prevents the cells from knowing when to stop. And the minute that you break that checkpoint, what happens is that cells normally will grow until they touch each other. They rest against each other and then they basically stop growing. if you delete one of these checkpoints, you end up in a situation where the cells touch each other and they're like, oh, well, there's no reason to stop. And so they grow and they grow and they grow, and then you get a tumor. And so transposons, if they jump into one of these regulatory genes, can cause a lot of problems. But if they're really 20% of the human genome, which is it's a hypothetical estimate based off of these inverted repeats. And so it could be much too high, right? There's not a guarantee that that's an accurate number, because people have just, by bioinformatics, looked and identified these repeats. And they're like, well, it must be a transposon, because these are the repeats that transposons use, but that's not necessarily a clear and direct line of evidence for right now.
Humans have heads, but not everything with a head is a human.
And so what's weird is that transposons are basically like protoviruses, because when they're transcribed, they make themselves, they first of all are able to transcribe themselves more because they make a protein that transcribes them and then makes them into, it's a reverse transcriptase actually. So what it does is it takes the mRNA that's made and then it makes it into DNA, and that DNA is what gets inserted back. And so it's basically this kind of self-promoting molecule. And there's also...
And there's a lot of, I was reading a bit about the theory behind this, and people think that these have been really important in generating the genomic diversity within populations necessary to spur new adaptations in the population. So there's like an evolutionary basis for why your genes would want to be doing this.
You want to have as much diversity as possible. And so it does make sense that your genes would want to be doing this, but it's a little bit, so I guess transposons, it's a little bit tricky because you don't really carry a lot of other genetic information with you.
Well, you do from viral interactions, right? But
not from transposons. Like transposons are particularly strange because if you look, this contains an open reading frame, and that contains this reverse transcriptase and an endonuclease, which is the scissor that's able to actually cut this DNA. But they often don't carry a ton of other information with them.
I think this paper is making the claim that it does carry fragments of the SARS too.
And in this case, what they are saying, if we go back to the paper...
So it just makes me wonder if so much of the reason that they've persisted in the genome in general is because they're associated with other viruses that over eons have accumulated in the genome as well.
And so basically what they did is they took these, in their experimental setup, they took a cell line which had this line 1 expression. They infected it with SARS-CoV-2. They waited for 2 days and they took the RNA, they extracted the RNA, cut it up and sequenced it. And what they found is they found all of these human COVID chimeric reads that were basically telling them, sorry guys, that they basically found reads that had human DNA on either side and a SARS-CoV-2 piece of DNA in between them. And what they pointed out was they were like, if you really do view this as being a transposable element, you would expect to find it periodically distributed randomly through the genome of the cells. And that's exactly what they find. It's like it's not always oriented in the exact same direction. Sometimes it's oriented forward, sometimes it's oriented backwards. Transposons don't have a direction associated with them. They're elements that allow them to jump on either side are mirrors of each other. And so they can jump in one way or another way. And that can be better or worse depending on what it is that your transposon is jumping into.
One more thing that I think makes it kind of, this is, I think, one of the more compelling arguments about this being significant, they also say here, is that they looked at some patient samples and they found in a few of the patients anyways, some of the patients, the majority of the viral transcripts that they sequenced appeared to be derived from integrated viral sequences. So they found host DNA transcript pieces in them, or transcripts from the DNA in them as well.
Sorry, I didn't follow. Say one more time.
I'm just looking up here. Yeah. You're there. Yeah. So they looked at some patient samples and they found that a lot of the transcripts, the viral transcripts, had pieces of the host in them as well.
And this makes sense because they weren't actually active virus. Like these cells didn't contain within them a full virus that was able to be popped out and could infect somebody else. Like if you look here, it has basically a chart where, as far as I can tell, it's only this little piece of the SARS-CoV-2 virus on this N-terminal end that's jumping in. And so it doesn't seem to be some massive thing. Like if you look at this is a really
might be
small.
Enough to test positive by PCR or something. That's the
It
question.
may be, but it's not of the spike protein. It's not of the spike.
Right, it's not like a
subunit.
virus, right?
Yeah, so it's not coding anything. It's just kind of a proof of concept that in the presence of a transposable element, there are aspects of SARS-CoV-2 that seem capable of jumping into the somatic, the DNA.
So, how cool and terrifying is that? Anyways, yeah, like Dr. Benenberg said, it doesn't appear to code for anything, but of course we wouldn't know it did until we saw that it coded for something. And this is just kind of how the genome evolves over time anyways. This seems to just be another instance of it. We know that a huge portion of the genome is made up of viral DNA, and the thought has traditionally been like, oh, well, those are probably from DNA viruses. And it's like, well, maybe not all of it.
Yeah, I mean, they also make the case for at the very end in their conclusions, they're basically saying that, the number of people that this is actually happening in, or the number of cells that this is happening in, could be vanishingly small. I mean, the margin of error is enormous, right? It says it's been estimated that only between 1 in 1,000 and 1 in 100,000 mouse cells infected with these viruses, either in culture or if the animal actually carried the virus, that it was integrated into the genome. So they're looking at a different virus, and they're basically saying, well, look, if you look at this other one, even when we do mouse studies and we look to see if it's integrated into the genome, we don't think that it's happening that often. And
well,
so
they also say the incidence is enormous, though. I'm trying to find this.
Which is the next statistical part. It's exactly. They're like, look, if you have 140 million humans that are infected with SARS-CoV-2, then even if it's 1 in 1,000, it's pretty big pretty fast. If it's 1 in 100,000, then, you're not going to have that many, but the range is wide. And so this is very preliminary evidence that does seem to suggest that it's possible for these long COVID symptoms, when you're not really positive for active virus, could be due to the fact that this transposon is integrating into some part of the genome that is causing these widespread symptoms.
I wasn't even going to go that far, personally.
Really?
I mean, maybe. I was thinking it might just be more like, okay, well, first of all, it's really cool and interesting by itself. And I was thinking it might just tie into the fact that that they're having these fatigue symptoms, which are correlating with them testing positive or not. And why are they testing positive when they're not infected? And does it mean that they're still infected, or does it just mean that they've integrated some piece of it into it's also one of the first, I don't know, maybe there's other examples, but it's one of the first ones I've come across, which doesn't mean that much, but one of the first instances I've come across of this viral integration of an RNA virus in an interesting way, because I've always heard the idea that, okay, well, our genomes are mostly viral DNA, and clearly that comes from DNA viruses, but looks like that's not necessarily the case, which is cool. So all viruses, equal opportunity employer for your body.
I mean, I think this covers what I have to say about this. Should we go to questions?
Yeah, let's stop there. Yeah.